Hello, welcome to North Ronaldsay Science Festival. My name's Alex Wright. I live on North Ronaldsay, and today we're interviewing with my friend Renee, who's a PhD student in Copenhagen. I'll pass you over to him.
Okay, thanks Alex for getting in touch with me. So yeah, I'm going to talk today about plastic waste, recycling and bio-valorisation of said plastic waste. So first, as we all know, plastic waste is a huge problem. So for example, just some stats for you, in 2015, 380 million tonnes of plastic was produced globally. Of this, 141 million tonnes of that was turned into plastic packaging waste. So plastic packaging waste is mostly in the form of polyethylene terephthalate, which I'm going to talk a lot about later. 45 million tonnes of it was textile waste. And a surprising fact that I've learned recently is that textile waste is majorly from discarded fishing nets, which are then left to float around in the ocean, probably catching more fish as they go. And yeah, in Britain alone, 1.7 million tonnes of plastic waste is produced per year, or was in 2015. So that's equivalent to around 800 times the mass of the London Eye. So it's a huge amount of plastic. And we also in Britain produce 1 million plastic bags every minute. So a study was done recently on the amount of plastic waste produced since pretty much the start of plastics being used in society in 1951. And it was found that 8.3 billion tonnes of plastic has been produced. Of that, 7% of it has been recycled into new plastic products, and 10% has been incinerated. So when you do the maths on that, you can see that there are an estimated 800 kilograms of persistent plastic waste per person currently alive on Earth, which is a huge problem. And of course, it's the persistence of plastics waste, which is the issue. So plastics, their main properties which are useful, which make them useful to us, is their resistance to degradation. That's why we can use them to preserve food and make strong buildings and strong products that are going to last. So what happens to these persistent plastic wastes that we have? So in the developing world, a lot of them end up in landfills where they can leach into groundwater and where they can make land unusable for future generations. And in the developed world, often they are used, plastic waste are incinerated to produce energy. So while this may sound like a good thing to produce extra electricity, obviously the problem is that it produces a lot of carbon dioxide, which is pumped into the atmosphere. But more than both of these two things, most of our plastic that we currently waste ends up in the ocean, which is obviously a big problem for marine life and for conservation of that. So in each ocean around the world, there are huge garbage patches of plastic. So for example, in the North in the North Pacific, there's a garbage patch that is over 1.6 million square kilometers in size, bigger than the US state of Texas. This is mostly made up of microplastics, which are produced from large-scale plastic products being thrown into the ocean. And then these weather under constant UV light and oceanic currents to break down into smaller fragments, into microplastics. These are just defined as plastics that are smaller than 5 millimeters in diameter, and they can persist for up to 500 years in the ocean. So this is obviously a problem for ocean life. So it's been shown that 70% of deep ocean fish have eaten plastic, and maybe more problematically for humans, 55% of all the fish species that we eat are found with microplastics in their gastrointestinal tract. So what can that do for human health? So obviously, if they're in the fish, they end up in us. The microplastics can concentrate up the food chain and then make their way into the human body. And of course, it's not just people who eat fish that have this problem as well. Microplastics are found in pretty much every human body. So it affects things like cell respiration. This is a process that's affected by microplastics. And also the immune system is affected by small plastic fragments in the body. It recognizes them as foreign agents and then tries to remove them by the use of the immune system. But then this leads to white blood cells being clogged up with tiny fragments of plastic, making it less able to fight off new infections the next time a real infection comes along. Plastics themselves are hydrophobic, meaning they don't bind with water, but this also means that they bind other hydrophobic materials, such as heavy metals or certain toxic chemicals used in production processes, and these can often be carcinogenic. So as the hydrophobic materials bind to the plastics, they make their way into the human body and can cause a lot of problems. So yeah, they've been found everywhere in the human environment. For example, microplastics have been found in German beer and in Korean shellfish, and also in human stools, showing that they're already within our body. And sadly, they were also found last year for the first time in the amniotic fluid of a pregnant woman, so showing even unborn babies are being affected by microplastics. So what is to be done with all this plastic waste that we have? So for example, here in Denmark, where I live, 57% of the 350,000 tons of plastic waste produced in a year is incinerated. So while this does produce a lot of electricity, meaning lowering the reliance on fossil fuels, it does mean that over 1 million tons of carbon dioxide are added into the atmosphere because of this incineration. So 13% of the plastic also produced or used in Denmark is recycled here in Denmark. And this is done by very energy intensive means using high temperatures and high pressures, and can often also use the required use of toxic chemicals such as ethanol or methanol, which are then wasted into water streams. Also, these high temperatures and pressures also need to use a lot of electricity, therefore are less sustainable than some of the other things I'm going to go on to talk about in a minute. And unfortunately, the problem with this as well is that the plastic produced after recycling is not the same grade as the original virgin plastic used. So this means, for example, recycled PET, polyethylene terephthalate, cannot be reused to package human foods. It's not of a high enough grade. So it needs to be used for lower grade things, like a lot of fabrics in carpets, for example, are made from PET these days. So because of this unsustainable way of recycling, it leads us on to biodegradation. So this is done by many organisms that I'll talk about. And because living organisms do it, it takes place at much lower temperatures and at normal atmospheric pressure. And it reduces also the need for toxic chemicals. So there are several organisms that degrade plastics. So this first one here, the Galleria melanella, is a wax worm moth that can degrade polyethylene. So it literally eats it and then excretes the products afterwards. There's a mushroom that grows in a lot of Northern Europe called Pelastopsis microspore. This degrades polyurethane. So again, a organism in the environment that we all live in can degrade a plastic. But the thing I'm going to talk about most today is a bacteria called Ideonella sakaiensis. And this degrades polyethylene terephthalate, the widely used packaging plastic. So the bacteria itself was discovered in 2016, so it's a pretty recent discovery. And it was interestingly isolated from the waste outlet of a plastic recycling factory, which obviously has a lot of microplastic fragments in there. So it's interesting that in this environment, a selective evolutionary pressure was made. There was a lot of microplastics in this water, and then that evolution has retooled already existing enzymes to degrade a completely new material. So a material that has only been in the environment since the 1950s, something has now evolved to eat it. So that's quite incredible. And it shows that everything is food for something one day. So the bacteria itself converts PET into biomass, into new bacterial cells, but also into carbon dioxide. So around 60% of the plastic is converted into carbon dioxide, which again is a problem. And another problem with this is that the bacterial colony can only degrade one piece of microplastic in around a week. So it's very slow acting, and the side products it produces are not ideal. But nevertheless, it's a major topic in current research, and it's got a lot of people excited. So what people do, there's some of the research they do, is they take the enzyme out of the bacteria and use that to degrade plastics outside the cell, thereby removing the problem of creating carbon dioxide. So they take this enzyme here, PETase, which is polyethylene terephthalate hydrolase, and it breaks down PET, it breaks the bonds in PET just using water, nothing else. So it breaks down the PET into an intermediate product, which is broken down by a second enzyme, also from the Ideonella sakaiensis, and breaks it down into these two products you can see here, ethylene glycol and terephthalic acid. So these are the two monomers that are originally used in all cases to make the PET plastic in the first place. So these can then be repolymerized into new PET, which is of the same grade as PET that is made from fossil fuel sources such as oil. So that's a very exciting way to recycle the plastic into new grade plastic, also done at 30 degrees C, so uses a lot less energy. But so as we said, we don't want to do it with the original Rennison saccharensis. So the genes for these two enzymes are put into E. coli cell factories. Then these produce many much higher concentrations of the enzyme than the Rennison saccharensis would. Then the enzyme is purified and put to use at large scales to degrade large amounts of plastic. So that's the general usage of the enzyme, but I'm going to talk a little bit about the project in particular I'm working on, which is an EU-funded project called Disrupt. So yeah, it uses this circular economy kind of system in which plastics are broken down into their monomers, such as by enzymes such as PETase. But there are many other enzymes that are known that can break down other plastics into their monomers as well, taken from the other two organisms that I spoke about previously. So these monomers are then fermented to produce organic acids. So this is done in a similar way that when milk, for example, is fermented to produce acids that help the milk curdle into cheese. So in the same way, these monomers from the plastics are fermented with especially engineered organisms, specially engineered strains of E. coli, and they produce some lactic acid, but also different organic acids. These can then be made into biodegradable plastics and new plastic products, which are easier recycled next time. So, for example, the polyethylene terephthalate you see there on the left is broken down using PETase into these monomers that you see in the middle here. Then we do a fermentation with a specially engineered strain of E. coli, which will produce these two products on the right, so lactic acid in the same way that the lactic acid cultures that are used in making cheese, and the furan dicarboxylic acid. So these two organic acids can then be repolymerized into different plastics, such as polylactic acid, which you've probably seen in a lot of the recyclable cutlery that you might get in restaurants and things. That's often made from polylactic acid. And polyethylene furanoate, which has a lot of the similar characteristics as polyethylene terephthalate, but it has the important characteristic of being biodegradable. So the project essentially uses the same raw material of current non-renewable plastics, such as PET waste or polyurethane, to make new renewable plastics that will either be easily degraded by less specialized microorganisms in the ocean, if that is where they end up, or they can be collected and more easily recycled in this circular economic fashion. But of course, this is not the only project happening. There's many other projects being funded by the Horizon 2020 EU funding project. And in the industrial sector, there's a company called Carbios, which is currently reporting that they can break down a single plastic bottle in under 10 hours using enzymes at temperatures of around 30 degrees C. So it's a bright future for breaking down plastics with biodegradable models. So yeah, that's me. I'd just like to say thanks to the British Science Week for funding the North Rennison Science Festival, and thanks Alex for asking me to talk.
Thank you so much, Rene. That was so interesting, and it's especially relevant to here because so much of the waste we go and collect on our litter picks along the shore is actually fishing waste. It's nets and buoys, and
so fishing waste is just discarded by fishermen and left to float around the sea.
Yeah, and we see a lot of it. It gets washed up on our shores. So how long do you think it'll be until your process is commercially viable?
So it's around it's a 5-year project that we're doing. So by the end of that, we hope to have a proof of concept for a mid-level scale degradation of non-renewable plastics and turned into new plastics. So within around 5 to 10 years, it should be on a commercial level able to make money and recycle these plastics into new products.
Oh, thank you. I was thinking about the fun time we had last time I was in Denmark when we were trying to break down these plastic bottles using a food blender, and it wasn't particularly brilliant. Have you improved that process now?
Yeah, that was quite bad. We were like pushing it in with a wine bottle, I think, into a blender, and yeah, so it was a bit dangerous. Yes, we have in the lab now a sort of industrial blender that can break down PET, especially into small fragments, which are then more efficiently degraded by the enzymes. And we're also using some like low-level heating techniques, so up to around 60, 70 degrees, which can then break down the crystal structure of the PET plastic and make it again more easily accessible to the enzymes for breakdown.
Brilliant. Oh, thank you so much, Rene. Thanks so much for doing this. And I'll say goodbye now. Bye-bye.
Thanks, bye.
Bye-bye.